In this video, we're going to do the second half of our brief tour of the history of microbiology and the microbiologists who led to the understanding that we have today. And we'll pick up now with Louis Pasteur's experiments, where he used the swan neck flasks. And there was another, this time British, biologist named John Tyndall, who extended Pasteur's experiments in the 1870s. And he was able to thoroughly replicate the work that Pasteur had done because he developed techniques and methods. He recognized the role of endospores, and he developed techniques that dealt with the existence of endospores in the experimental issues that some of Pasteur's colleagues were having. And he replicated Pasteur's experiments and also his own through a process that he was eventually dubbed Tyndallization. So here's John Tyndall, and we can see the experimental setup that he used for replicating Pasteur's experiments. So he built a box that could be completely sealed, and he coated the interior, so he wiped the interior of the box with a super sticky substance. And then he could set up his test tubes with broth in them, and he built a mechanism that he could operate from outside of the box to remove the lids from them. And so he put everything all together, and then he waited for some period of time until the light was what he described as optically pure. So he could shine light through it, and there was no shadow cast by any motes of dust or whatever in the air itself. And so he gave time for the air to settle, and the sticky insides of the box then trapped the dust and any other materials that might be in the air, and made the air essentially pure by allowing gravity to do that work. Then he triggered the mechanism that popped the tops off of the test tubes, and he could show that his thoroughly sterilized broth did not become a culture medium for bacteria. So Tyndall was the one who very securely put the last nail in the coffin for the spontaneous generation ideas. And so now we're left with, okay, so without spontaneous generation, what do we know about the causes of diseases? Okay, so now we got to look back in time once again. So back in Italy in the 1500s, mid-1500s, Girolamo Fracastoro, I'm not sure how you pronounce his name, but I think I'm somewhere in the right zone with that. He first proposed what we now talk about as the germ theory of disease. And it was his thesis that microorganisms, what we think of as germs, are capable of invading other organisms and causing disease while they're there. And so the work that Pasteur and others were doing in the late 1800s and early 1900s finally provided convincing evidence for the germ theory of disease as well, finally settling that matter, that it wasn't bad air that caused disease, it was instead the germs that were in the air or that were being carried by insects in the air that were causing those diseases. And in fact, if we think about the disease malaria, mal is bad, area, bad air was considered the reason or the cause of malaria. We now know that it's actually a protist that is transferred by the bite of mosquitoes into the blood of humans. So with Girolamo Fracastoro's work, and then the final work that was done by Pasteur and his compatriots in the late 1800s, we now recognize that for every disease there is a causative agent. So just to finish up talking about Pasteur, because he was so important to microbiology, Pasteur and his colleagues at the Pasteur Institute were responsible for a number of interesting discoveries and explanations. The first one, Pasteurization, is a process that we now use in a lot of food production especially, or anything where we're trying to create or produce something that is reasonably sterile. So pasteurization is a process of very quickly raising the temperature of a fluid to a level that kills bacteria or kills the microorganisms that are in it, or kills most of them at the very least, without altering the flavor of the material or other qualities of the liquid that we're trying to make reasonably pure for consumption or other uses. And pasteurization, well, Pasteur came up with these processes because he recognized that sometimes wine turned out really well. So some yeasts make really good wine, and remember, it's yeasts that are responsible for making the alcohol as wine ferments, while others produced wine, other yeasts produce wines with kind of an off flavor, kind of an oily taste to them. And so what he did was to heat the grape juice to a reasonable temperature that could kill things, kill microbes, with the lack of oxygen around, and then into this now sterilized, pasteurized product could introduce a good yeast that would consistently and over time make very, very good wine. So that's pasteurization. We now use it with milk and other products that we need to keep reasonably sterile. He also established the link between microbes and diseases that occurred in silkworms. So people were growing silkworms in order to harvest the silk from them. And here I show you, here's a silkworm moth, here is the larva, the silk quote-unquote worm, and here is the pupa, the stage from which the silk strands are actually harvested. And with this connection between a disease in the silkworms and the microbe that caused it, we see the first time that linkage between a microbe causing a disease. And this was a foundational piece of work in developing and establishing the germ theory of disease. And finally, he developed and tested the first Western vaccines using the dried spinal cord of rabies-infected rabbits. So they introduced rabies into the rabbits, and then they could harvest and dry the spinal cord, which is where the virus that causes rabies is kind of has its most damaging effects, and then could grind that up and introduce that into human subjects or other mammals and use it as a vaccine against rabies. And he was following a process that had been used for centuries in Asia. So here we are again at that place where this textbook is just super Western centric. So in Asia, India especially, they have been using these kinds of techniques for centuries to protect against disease. But Jenner and Pasteur are the ones who discovered vaccination. Okay, so now we'll move on to Germany in the late 1800s. Robert Koch, K-O-C-H, you say the C-H as a K sound rather than a Ch sound. So Robert Koch, a contemporary of Pasteur's, he was studying bacteria and disease, and he discovered that Bacillus anthracis, it's a bacteria found in it's cosmopolitan in its distribution, found just about everywhere, and it forms endospores, so these resistant structures. And he discovered Bacillus anthracis in the blood of cattle with anthrax. And so he isolated the Bacillus anthracis from the blood, created a pure culture of it, and then went and injected it into healthy cattle. And what he found was he could induce anthrax in those cattle. And so using this technique, what we come now to refer to as Koch's postulates, he was able to very convincingly show that Bacillus anthracis is the responsible microbe for causing anthrax. So here are Koch's postulates, and another view of Koch too. He looks just so darn thoughtful. Koch's postulates are the scientific method that's used to establish what is causing a disease. And so the first postulate is that for every disease, there is a specific causative agent. And in order to understand the disease, we have to find that causative agent. the second postulate, that in order to confirm that this is the disease-causing agent, first the disease organism has to be isolated and grown in pure culture, so that there are no other microbes growing in the culture medium, so that we know that we've isolated just the causative agent that we're investigating. Third, to establish that it's causative, we have to be able to inoculate a healthy but susceptible animal. So sometimes animals have encountered a disease before, have survived it, and now they have developed immunity to it. So you have to find an animal that is susceptible to the disease and inoculate it with the pure culture and demonstrate that that culture is now triggering the same disease that we're investigating. And then fourth, you have to be able to show that the disease organism is found in the inoculated animal, so that we can, again, confirm the causal link between the disease organism and the animal. So, first, every disease has a causative agent. Find the agent and culture it in pure form. Third, inoculate an animal that can get the disease with the agent. And then fourth, after the animal develops the disease, re-find the agent in the animal's tissues or blood. Okay, so those are Koch's postulates. So some other contributions that Koch was super important, as you can tell, not just in identifying diseases, but in the work that he and his associates did in other realms of microbiology. So Koch and his lab assistants developed many of the culture and isolation techniques that you're going to learn in this class. They developed the use of agar as a medium to grow bacteria upon. This came as a suggestion at a dinner party from the wife of one of his assistants. They were looking for something that wouldn't melt at higher temperatures. They were using gelatin, I think, at the time. And the wife said, hey, I make these gels to eat in my kitchen. I make these gels using a material called agar-agar. It is ground up, or it's isolated. It's a protein, or it's a polysaccharide that is isolated from red algae, and it was used in cooking at the time, and still is. You can find agar-agar at the grocery stores. So, if you can find a specialty store, and so the wife suggested, hey, use some of this agar, and it won't melt at higher temperatures the way that gelatin does. Also, petri dishes came from Koch's lab. One of his lab assistants, whose name was Petri, developed these two halves. These dishes which had a larger dish that fit over the top of a smaller dish, so that air could still get to the culture, but dust and microbes were prevented from coming in. And finally, they developed the protocol to very dependably isolate individual microbes so that you could create pure cultures. The French, Pasteur and his colleagues, used a process of just diluting and diluting and diluting, so doing what are called serial dilutions of a sample, where you dilute a sample, take a little bit out of it, dilute it again, take a little bit out of it, dilute it again, and then hoping that at the end of these series of dilutions, you have only one individual bacterium in the culture medium that you have. That was the French technique, and it did not guarantee pure culture. Whereas the technique that you're going to learn in lab is one that allows us to guarantee pure culture. Okay, so those are some additional contributions of Koch and his colleagues. And then we'll wrap up here with the attempts that were made at infection control in hospitals at the time. So infection control infections in hospitals were huge, and you'll understand why when you hear my story. A guy named Ignaz Philipp Semmelweis. So Semmelweis was an Austrian working in the mid-1800s. So we've gone back in time just a little bit once again. And if we can believe this picture, he's a dude with an extremely enormous forehead, maybe a 5 head there. So Semmelweis recognized that there was a connection between a condition that women developed called childbed fever, also called puerperal fever, and poor sanitation. Because in the hospital that he worked in, the doctors would go from autopsies in the morgue in the basement of the hospital upstairs to the birthing rooms without ever washing their hands. And so they would have had their hands inside dead bodies of people who had died of who knows what, or whose bodies were beginning to decompose, and they wouldn't bother to wash their hands. And then they would go and examine the women during childbirth, putting their hands, looking inside the vagina and assisting perhaps even with labor and delivery without ever washing their hands. Remarkable. They would probably wipe their hands dry. It wasn't like they went upstairs with like goo on their hands, but they would have just wiped their hands dry rather than using some sort of soap to clean their hands. So he began requiring in the hospital that he worked in doctors to wash their hands with a chlorinated solution. So he used chlorine, or which was known to have antibacterial properties. He used chlorine in the hand washing solutions. And what they found was an immediate drop in death due to childbed fever, this puerperal fever. And even though he was successful, and this is just unbelievable to imagine, even though he was successful, he was ridiculed, he was harassed, eventually wound up in a institution, and I may be getting him confused, we'll see. But I think he was the one who died of the same puerperal fever. Okay, so that brings us now to Joseph Lister, also very important in infection control. And if you recognize the name Lister from Listerine, so using some of the same chemicals that used to be, I don't know that they're using them anymore in Listerine, but used to be used in the production of the mouthwash and Listerine. So Joseph Lister, English dude, 1865, he read the work of Semmelweis and of Pasteur. He recognized, even though it had not been firmly established yet, that link between sanitation and disease, and he began using carbolic acid, which can easily be generated by using this solid form, putting it in water, and then a chemical reaction takes place, and you get a vapor of carbolic acid that pours out of the container. And so he used this carbolic acid to sterilize the bandages and all the surgical instruments that were in use in the hospital where he worked. He also would mist this carbolic acid across the surgical theater, and during the surgeries he would be using this carbolic acid to help maintain a sterile area. He was ridiculed, but his recommendations were eventually widely accepted. So Semmelweis died, died in shame with the ridicule. But Lister, even though his early requirements were being ridiculed, later people got on board with it. And so we consider Lister the father of antiseptic surgery because of all the work that he did to promote antisepsis in the surgical environment. Okay, so there we are at the end of all of our little bits of history. Well, I say that. We're going to wrap up in the next lecture our history by talking about the golden age, the quote-unquote golden age of microbiology. But we'll call it quits here with this one, with this conversation. So, as always, if you have any questions, you know how to reach me.